Hi, Gen Bio. We're back now for week 2 of our spring semester, where we're going to look at deuterostomes. And deuterostomes, the two large groups within that classification are echinoderms and chordates. Okay, so this phylogenetic tree should look fairly familiar to you because we saw it last week when we were talking about protostomes. Okay, so just to sort of orient ourselves, everything we're looking at today is going to be either an echinoderm or a chordate, and these are classed together as deuterostomes. So again, they are all in the animal kingdom. They have true tissues. They, at least at some point in their life cycle, have bilateral symmetry. They have body cavities that we had referred to last week as a coelom, the space that is in between the digestive tract and the body wall. So that coelom, again, is enclosed by mesoderm, which is the third germ layer that everything in this lineage has also. So another way of saying that is that they are triploblastic, 3 germ layers, the endoderm, ectoderm, and the mesoderm, which encompasses that coelom. Okay, and then last week we looked at protostomes, right? We looked at the Nereus and the crayfish. And this week we're going to look at deuterostomes, which are in the groups echinoderm. For the echinoderms, we have a sea star for you to look at, and then chordates. So we have several different chordates that we're going to look at this week. I'll also mention that we had talked about a few differences in the development last week between protostomes and deuterostomes. So we'll flip to the next slide. One of the main things that we had talked about is that the blastopore becomes the mouth in protostomes, and the blastopore becomes the anus in deuterostomes. So again, just to refresh, as a cell gets fertilized, it then undergoes cleavage, which is this period of rapid cell division and little cell growth, but lots of division, which will result in this ball of cells, which then will have an indent that will start to form. It's called the blastopore. And that blastopore extends into the archenteron and ultimately extends throughout the body. And that will be the animal's digestive tract, mouth to anus. And so for protostomes, that first indent, that blastopore, that's going to become the mouth. The ones we're looking at today, deuterostomes, that first indent will be the anus, and then the mouth will be the second opening that is formed through extension of that digestive tube. Okay, so that was one major difference we talked about last week. We also talked about the formation of the mesoderm. In protostomes, that mesoderm sort of starts to form on either side of the blastopore, versus in deuterostomes, the mesoderm starts to form from extensions, little buds off of the archenteron, and then it will sort of migrate. These cells will migrate around and form this mesodermal layer. Okay. Another difference, which we didn't talk about last week, is that during cleavage, there's a difference in how the cells divide between the 4 and the 8 cell stage. So obviously, upon fertilization, that one fertilized cell is going to divide into 2, is going to divide into 4, is going to divide into 8, right? But at the 8 cell stage for protostomes, the little ball of cells is not exactly having one cell stacked on top of the other. So you had 4, and then 4 of them formed by division of the first 4. And the orientation was sort of shifted, like catty corner a little bit. And we call that spiral cleavage. Whereas in deuterostomes, the cleavage occurs between the 4 and the 8 cell stage, and those second 4 sets of cells are stacked directly on top of the first 4. So that's called radial cleavage. One other big difference at this point is that The cells in protostomes, even at this stage, their fates are determined. So we call this determinate cleavage. So if you take one of these little cells and isolate it, its fate was already determined. It's already sort of programmed to become a specific portion of this organism. Whereas with deuterostomes, cleavage at this point is still indeterminate. So what that means is that you can take this single cell and it has the potential to develop into a number of different structures in this organism. So its fate is not yet determined. This is actually what happens when identical twins are developing. There is a split in the developing ball of cells, right? And then that split has the potential to develop into, in fact, an entire other organism. So this is called indeterminate cleavage at this point for deuterostomes. Okay, so that's a difference we hadn't talked about. Everything we're going to look at today is in this category, these deuterostomes. Okay, the first organism that we're going to look at is the sea star. And it is a model that we're going to look at for the phylum Echinoderma, which means spiny skin. Echino is spiny, derma is skin. And that makes sense because if you feel the sea star, it does have a lot of spines all over it, okay? And it's in the class Asteroidea. It is classified as a deuterostome, which, if you recall, puts it in this bilaterally symmetric branch of our phylogenetic tree, right? However, looking at it, you could make a pretty strong argument for the fact that it has a lot of characteristics of radial symmetry, right? If you cut it on various planes, it looks very much like it has radial symmetry as an adult. However, it is classed in the bilaterally symmetric group due to a few factors. First of all, if you think way back to last semester, we looked at embryonic development in slides using sea star, right? We looked at cleavage, we looked at the blastula forming, the gastrula, and the bipinnaria larva, right? And if you notice at that stage, there was actually bilateral symmetry. As an adult, it tends to take on more this radially symmetric appearance where you've got the central disc in the sea star surrounded by 5 arms, usually 5, it can vary. But so as a developing larval stage, it exhibits bilateral symmetry. It also has 3 germ layers. And so that for those reasons, it's classed with the bilaterally symmetric organisms, even though as an adult, that's kind of a little unclear. So this is our, like I said, our model organism for the echinoderms. And since it, as an adult, looks pretty radially symmetric, we sort of distinguish the surfaces now based on the top and the bottom side, right? So the dorsal and the ventral side, or in this case, we call it the ventral side here is the oral surface. And the dorsal side here is the aboral surface. And that is because on the oral surface, there's a mouth, okay? And that's shown right here in the center of the central disc, okay? And then there are these arms that radiate out from the central disc. And these have on the oral surface spines on the outside and then tube feet on the inside of each of the arms. These little structures here are called tube feet, and they help with the movement of the organism. They're also a part of the sea star's water vascular system, which we will talk about in a few minutes. But first, we're going to look at then the aboral surface. So just some external anatomy. In the middle, like I said, is the central disc. That's shown here as like number 3 in the picture. I'll enlarge it a little bit. Number 3 in the picture is the central disc. Number 1, you'll notice, is a little spot. It's sort of an off-white cream color. And if you touch it, it's got this very sturdy structure to it. When you come to lab, next week, we're going to have these organisms for you. This is the Madriporite. It is the entrance to the water vascular system for the sea star. So water is able to enter here, and then it will travel through different tubes and passages and help with several different functions, one of which is movement, another is respiration. So that is the entrance, which you'll see. It's called the Madriporite. There are spines that cover the entire surface, and each of the extensions from that central disc are called arms, or sometimes called rays. Okay. So we are going to turn to the next slide, where you can see a lot of the same structures that I just discussed: the Madriporite, the central disc, the spines, and the rays or the arms on the oral surface. The oral surface again has the mouth, which is number 5 here. This is on the oral or ventral surface. It's usually facing down. And then these 2 feet, which are in the center of each of the arms. They're located in what we call the ambulacral groove. Okay, so this is a little groove inside each of the, sort of towards the inside of each of the arms. And the 2 feet, if you feel, are definitely softer, much softer than the spines, which are sort of on the outside of each of the arms. Okay, and then this is what the oral surface looks like normally. However, sometimes you can actually see the stomach protruding from the mouth. If you look here, you can see that in this picture here. I'll enlarge it again a little bit, showing you that the mouth is just this opening, but at times the stomach actually protrudes through that. And this can be extended to help engulf prey for the sea star. Okay. All right, so that pretty much sums up the external anatomy that we can see in these pictures. Now, I don't have it pictured here, but when you are in lab, you should also look for little eye spots on the tips of each of the arms. They're very pale, very small bumps that are basically on the tip of each arm. They sense light for the sea star. Okay, so we'll look for those in lab, although they really can't be seen in the pictures. All right, so like I said, that is the external anatomy for the sea star. Now we're going to look at some internal anatomy. So what we're going to have you do when you come to lab next week is we're going to have you dissect the sea star by at first just cutting the tip off of one of the arms. So you're going to make a cut right here basically, and then you're going to cut up the sides of each of the arms. So, well, up the 2 sides of one arm, so that then you can sort of make your way carefully to the central disc. We want you to start at the tip so that you don't disrupt the features that we're going to be able to see in the central disc. Okay, so you're going to start by cutting the tip. You're going to cut up and remove the spines that are on the outer covering of the arm. And so when you do that, then you're going to be able to see several different structures that repeat in each of the arms. So the first structure that you will almost certainly notice is the digestive glands. Okay, so I'm going to enlarge my picture a little bit. Showing you that number 8 here, these structures, there's 2 per arm. You can see them kind of coming out of the arm here. Those are the digestive glands, and they kind of look like bunches of grapes. If you use your tweezers or a probe when you're working with a dissection, you can lift them out and you'll see that it kind of looks like a bunch of grapes, right? That would obviously not be purple, but they're like a palish yellow color. But in structure, morphologically, they kind of look like a bunch of grapes. These are the digestive glands. They help with chemical digestion for the sea star. And then if you remove the digestive glands, then you will see directly underneath another thing that kind of looks like a bunch of grapes. Okay, I'm going to enlarge the picture here and show you number 9 here. In fact, this one looks pretty clear here. There's a little strand right here. And again, it's smaller, but it has the same sort of shape as the digestive glands. It's a lot smaller, but kind of looks like a bunch of grapes. And there are usually 2 per arm. Sometimes they vary in size if it's more mature or less mature. They might be larger or smaller, respectively. But they would be located in each of the arms directly underneath the digestive glands. So that's number 9 there. So for attendance this week, my first question is, what did I compare the digestive glands and the gonads in the C star to as far as morphological structure? That's my first question for you guys for your attendance assignment for this week. Okay. All right. So moving on then, we will look towards the central disc now. Okay. In this area, this is where you're going to see the stomach. As we mentioned, sometimes a portion of the stomach can actually protrude through the mouth on the oral surface. But we're looking at the abral surface right now. We're kind of looking at the top side or the dorsal side of this organism. And so if you cut up and you remove a portion of the covering over the central disc, we're going to try to have you avoid cutting at first the location where the madriporite is, so that little bump on the surface. Avoid that to start with, and start to remove the covering on the other side of the central disc. And then what you'll see is that there's a stomach that has actually 2 portions to it. Okay, so there is a star-like structure that in this example, it kind of came off with the central disc, but you can see it has almost a star-like appearance where there's actually a connection running from the stomach there down and connecting into the digestive glands. The digestive glands are also sometimes called the pyloric cecae, or singular is cecum. And those connections I just said are called pyloric ducts. And they connect to the star-like structure, which is the pyloric stomach. Okay, so those, if you remember that word, the pyloric ducts connect to the pyloric cecae, or digestive glands, same thing. And they connect them to the pyloric stomach, which has a star-like appearance. And then just underneath the pyloric stomach is the cardiac stomach. And the cardiac stomach is more fluffy and kind of like a pillow, softer, more roundish and cushiony looking. And so you can see that here. These little bumps, kind of maybe like a flower almost, because there are 5 little round parts that extend into each of the arms, just a little bit. Okay, so that is the cardiac stomach. And then the cardiac stomach connects then to the mouth on the ventral side, on the oral side. Okay. I think that covers the digestive tract. We have the anus on the top. You can't actually see that, but there are digestive glands or pyloric cecae in each of the arms, help with digestion. They're connected to the pyloric stomach. Pyloric stomach sits on top of the cardiac stomach, which is on the oral side, just adjacent to the mouth. Okay, so that covers the digestive system, with one exception. There are also rectal cecae. So these are very small structures that I think it's shown right here. It's a little flap. It often comes off with the central disc. So we'll look for those as well when we do our dissection. And then if you look at this bottom right picture, now we're starting to look at the water vascular system. Okay, so we've covered the digestive system. Now we're going to turn our attention to the water vascular system. And like I said, the Madriporite is the entrance to that. So water will enter the Madriporite and travel through a series of canals, which bring that fluid throughout the central disc and then out to each of the arms and to the tube feet. Okay, so if you look here, you can see that the tube feet have a little bulb on top, on the inside that you can observe. These are called the ampulla, the top part of that tube foot. And on the other side, when you flip it over, then you'll see the extension in the tube feet. Okay, so again, water moving through the system helps the sea star move. As the pressure is exerted on the water in this circulation, this network, then that tension allows for a pressure and a regulation of that pressure in different areas of the arms and the central disc, allow for the slow sort of gradual movements that are characteristic of the sea star. So we're going to look at the different parts of the water vascular system. Okay, and although you can see them in the pictures here, the Madriporite leading to the stone canal, which leads to the ring canal, and the extension then out into the radial canals. You can see these here, but we're going to turn our attention to the next slide because they're a lot more visible when they're in diagram form. So like I said, water will enter the Madriporite, travel through the stone canal, around the ring canal, out into each of the arms through the radial canals, and then between the radial canals and the 2 feet, there is a very short lateral canal. And then from there, the water will be contained in the ampulla, which is the top part, and move into the tube feet, which you can observe on the outside then of the sea star. Okay, so those are the major parts of the water vascular or locomotor system for the sea star. This again has a role in locomotion. It also helps to bring oxygen to the sea star. Okay. All right, so that wraps up our talk on echinoderms. We just discussed a sea star as an example, as a model organism for echinoderms. Again, that's one of the phyla within the Deuterostome group. Now we're going to look at Chordates, which is another phylum within that Deuterostome group. Okay, so for the Chordates, we have several different organisms that we're going to look at. One is the Lancelet or the Amphioxus. It's a very old lineage in the Chordate phylum. We're also going to look at a Lamprey. We have the adult Lamprey in lab for you guys to look up, but you will not be dissecting that. However, we also have slides of the larval lamprey, which is called ammocoetes, or I've often heard it pronounced ammocoetes. I honestly have no idea which one is correct. Pronunciation is not my strong suit, but we have both the adult lamprey and the larval ammocoetes. And then we also have a perch as an example of a ray-finned fish. And then later on in the semester, we have a pig for you guys to dissect as an example of the mammalian group. So those are the major subphyla that we are going to examine. So we're going to start with the Amphioxus or Lancelet. Okay, it could be labeled as either one. And we are going to look at this. It's very small. So we're going to look at this primarily in slide form, microscope slides. So you'll notice that on the Canvas page, I have linked videos that I made of these slides that you would have looked at if you were in lab this week. You are welcome to examine them in person. It would probably be a good idea when you get back to campus. But for now, do reference those slides, do watch them. I do think they're helpful. So like I said, we're going to start with Lancelets or Amphioxus, but we should recognize that all of these organisms we're going to look at are in this phylum Chordata, right? And Chordata, the Chordata phylum has 4 definitive characteristics which make us guide us to group them together. Okay, so these 4 characteristics you should know are the presence of a notochord, which is a supportive structure, a dorsal nerve cord, so nerve cord or spinal cord located on the dorsal side, as opposed to last week we looked at the crayfish and the Nereus. They had their nerve cord on the ventral side, if you recall, right, towards their chest or their abdomen. In this case, now we have the location of the nerve cord or spinal cord on the dorsal side. Okay. They also have pharyngeal slits and a postanal tail. Now, I should note that these 4 characteristics are not necessarily present at all different stages of the organism's life. Some of them are only present during embryonic development. Okay, so for example, currently I do not have a tail, but yet we are all in this group chordates, right? Because during development, we developed with an extension behind our anus that would be very similar to other organisms' tails. So these are the 4 definitive characteristics of chordates. You should be able to recognize at some point in their life all of the organisms that we will look at from here on had each of these 4 characteristics. Okay, so we're going to start with the lancelet or the amphioxus. It is an invertebrate. It is a filter feeder. So what that means is it kind of like burrows into some sediment in the water. It will then just basically filter food, allow things to get trapped in its mouth region, and that will be the nutrient source for the lancelet or the amphioxus. It is in the subphylum Cephalochordata. So if you break this word apart, cephalo refers to the head, chordata refers to the notochord. Okay, so like I said, the notochord is that support structure that is one of the 4 characteristics of chordates. And the reason that it is classified as a cephalochordata is because that notochord extends into the head. And in fact, it extends beyond the nervous system structures. So in many other groups, you'll see that the brain, or a conglomeration of cells that make up the organism's brain, is the most rostral structure in that head region. Whereas in lancelets or amphioxus, you'll see that the notochord actually extends beyond the nervous system structures, more rostrally. Okay, and so that is where it takes its name, cephalochord. The notochord extends into the head in these organisms. So that's my second question for attendance this week. Why is the lancelet, or the amphioxus, same thing, why is lancelet called a cephalochordata? Okay, why is it a cephalochordata? So you should answer that. And then amphioxus, I just like to know what the words mean. Means both ends pointed or sharp at both ends. And so you'll see this is our amphioxus. Okay, so amphi, both. Oxus refers to being kind of pointed. All right, so this is what it looks like, which makes sense based on that name. All right, so we have some organisms for you to look at in lab, but again, you're not dissecting these. They're very small. So we're going to look at the amphioxus primarily in slides. Again, you should reference the slides that I posted on Canvas for you guys. But we're going to talk through them in the lecture here too. So this is the lateral view of the whole amphioxus or lancelet. And then this is an enlargement of the head region. And I think this looks very similar to your pictures in your lab manual. So if you look, you can see this is a cleared, basically, preparation. You can actually see the internal structures very well. So if you look in the head region, you can see that you've got some mouth parts, right? There's some little tentacles here called oral cirri. These are little finger-like projections, right, which help to trap food. If you recall, I said it was a filter feeder. So anything that might get trapped in there will be directed down through the oral cavity. And these tentacles are attached to the oral hood, which is the membranous structure that surrounds that opening. Okay. I'd like to also point out the notochord and the nerve cord. And if you look here, I'm going to make this a little bit bigger so you can see it a little bit more. The dorsal nerve cord is shown here. It's more of like a pinkish purple color. It extends here and terminates at this bulbous region. That's their brain. And then the notochord, the yellow pale structure, actually extends beyond that into the head. So again, this is a Cephalochordata. Okay. So notochord and nerve cord will then, from the head, run the entire length of the organism's body, basically, along that dorsal side, all the way down to the tail. Okay. So that's their support and nervous system structures. We talked about the mouth parts. Behind the mouth, then, more caudally, you've got these slits, right? These pharyngeal gills and gill slits. So water is going to travel through this region where oxygen is going to be extracted from the water for the organism. And then the excess water will travel through the atrium and exit through the atriopore. Okay. And then oxygen will be extracted again through the gills. Any food that goes into the oral cavity will be taken down and processed in the digestive tract here, which includes the hepatic cecum, this little protrusion you can kind of see behind the pharyngeal gill bars. Okay, so a lot of the digestion of glycogens and fats and storage of those is conducted through the hepatic cecum. And then as the food continues, it will travel down through the intestine. You've got some more absorption of nutrients from the food, and waste will exit through the anus. Okay, so that is the lateral view of the Amphioxus or Lancelet. We also have some cross sections in slides for you to examine. I have some pictures here. I also have the recordings of the videos on Canvas. The most rostral cut is here. And as you travel back through the body towards the tail, you come to a more caudal section here. So you should look at all of these and be able to identify the structures that are shown here, as well as in your lab manual. So I'm going to talk you through this. There's, of course, a place where food needs to enter, right? This is called the buccal cavity, or sometimes called the oral cavity. Okay, so this is their mouth, basically. And we had talked about in the lateral view that there are these cirri, these little tentacles, right, that are present here around that opening. Okay, so obviously in processing of the slide, they kind of became disjointed a little bit. But those little things here, those are the cirri, and they would have been attached to the oral hood here. And then you've got, as you move more towards the dorsal side of the organism, you've got some musculature present on each side of the dorsal end of this organism. So these are called myomeres. So the little bundles of muscles are myomeres, and they are divided by partitions called myosepta. And I will show you those a little bit clearer. You can see there's a nice myomere here with a myosepta in between. Okay, so those are their muscles. And then along the midline, again on the dorsal side, you've got this round oval type shape here. That's the notochord. Again, it extends into the head. Behind that, more dorsal to that, is the nerve cord, the dorsal nerve cord. And then you've got a dorsal fin present on the outside of the organism. So that's the most rostral portion. If you move a little bit back, the next most rostral will look something like this picture here, where now you don't have the opening any longer to the outside, right? Now you have this space, which is now called the pharynx. So this space is the lumen of the pharynx, and it is surrounded by gill bars here and gill slits here. And again, this is water is moving through this region and oxygen is being extracted. Food is also traveling through this region at this point. and moving down the digestive tract. You still have musculature, so myomeres and myosepta, your notochord, your dorsal nerve cord, and dorsal fin. So a lot of those structures are the same as the more rostral one. Then we're going to move to this third section where now we're more down towards the abdomen. Okay, so we've moved beyond sort of the head region and now more towards the abdomen. You can still see some of the pharynx here, but it has been sort of shrunken a little bit because there's some other structures that are present here in the abdomen region as well. You've got the hepatic cecum right here, and it's usually only present on one side. You only see one of those. Again, it aids with processing of glycogen and lipids, fats, and sugars for the organism. And then also, bilaterally, you're going to see the gonads. Now, this one is a female. We're going to be able to tell the difference between the female and the male. I'll show you that in the next slide. But for now, I'll just point out that you can see that within the gonads, you can see that there are these little circles here, okay? And this is what tells me that this is an ovary instead of the testes, because there are little eggs developing in here in the female, in the ovaries, as opposed to what you would see in the male. So we'll look at the comparison on the next slide. But you should be able to tell the difference between the male and the female based on the morphology of their gonads present here. Okay, so we've still got, like I said, the pharynx, the gill bars and gill slits, the gonads, the hepatic cecum, myomeres and myosepta, the notochord, dorsal nerve cord, and dorsal fin. All of that is kind of the same as what we had seen before. Okay. All right, and then if we look towards the tail, now you no longer have the gills, right? The gill bars and slits. So this is no longer the pharynx. Now it is the intestine. Okay, so we're further back. We're now processing and absorbing nutrients from the food. And so this smaller diameter structure that is in the same position as the pharynx is now the intestine. Okay, and just behind that is the notochord. Just behind or dorsal to that is the nerve cord, again, surrounded by some musculature called myomeres, divided by myosepta. Okay, so those are the cuts through, again, a female Amphioxus. We're going to compare it now to the male. Okay, so the male's on the left, the female's on the right. This one, as you can see, looks very similar to the last one that we saw, where you can see these little circles inside the gonads, right? Those, if you blow that up or if you increase your magnification on the microscope, you'll see that there are eggs developing in each of these little sections here. So that will tell us again that it's a female. Versus in the male, it appears to be more solid at the lowest magnification. If you increase, you'll be able to see that there's some punctate cells on the outside, or kind of fibrous appearance to some of the cells towards the inside. So that should indicate to you whether you're looking at a male or a female Amphioxus. Okay, so you should be able to tell the difference. All right, so that wraps up our talk about the Amphioxus. We're going to move now to the Lamprey, which is a vertebrate, same as you and I. It's in the class Agnatha, which means that it does not have a jaw. Okay, we are going to look at like an adult specimen, which looks a lot like this in lab, but we're not going to dissect it. We're also going to look at the larval form, which is called the ammocoetes or ammocoetes. I don't know how you say it. Which is going to be on, we're going to look at that in slide form for this week's lab. As an adult, it is a very interesting looking parasitic marine vertebrate. It is parasitic because as an adult, it attaches using its horny teeth to its prey. So it can attach to, for instance, a fish, and these teeth really grasp onto the outside of its food source. And then it has a tongue, which has, it's very raspy, and it will actually work its way through the outside layers of its prey, outside skin layers, and feed off of its prey's blood. So it is kind of a terrifying concept as far as an adult organism. It sometimes can attach to people if you're swimming in a lake. You could possibly encounter one of these. However, obviously we can deal with that. We can detach that if that were to happen. The fish obviously is not able to do so. It does not have arms or the ability to really detach the lamprey. So its defenses against this parasite are less than ours would be. But so I find it kind of interesting. It's kind of gross in a very interesting way. But these are their mouths. If you look at the outside of the lamprey, you can see some other structures, anatomical structures as well. So at the head, at the mouth region, we've got the buccal funnel. This should be buccal funnel, not buccal tunnel. Buccal funnel is its mouth region. Okay, so all of this here. Inside are the teeth. If you look on the top of the head, it's got a nostril. Behind that is a pineal organ opening. And this has the function of being able to sense changes in light. Specifically, it's believed to be able to tell the difference between different wavelengths of light, so visible light versus UV light. And this, we believe, helps the lamprey sense its depth in water, which is where it lives. Okay, so the pineal organ opening, and then those will just be singular structures on the top of the head. It does have bilateral eyes and then bilateral external gill slits. There's usually 7 openings, which are the openings that allow for the access to the gills, okay, for the lamprey. And then as you move down the trunk, you're going to come across an anterior and a posterior dorsal fin, and then a caudal fin on the tail. So that's the external anatomy of the adult lamprey, okay? They're very interesting creatures. We have some in lab for you guys to look at. They're really cool. But we're also going to look at the larval form, which again we call ammocoetes or ammocoetes. I will admit that it looks a little bit like the Amphioxus, which is the one that we just looked at. Right here is the Amphioxus. Here is ammocoetes. However, there is some additional specialization in the lamprey than in the Amphioxus. So what you should look for to basically tell the difference is that you've got a little bit more cephalization. You've got eyes here in the larval lamprey. You've also got some increased specialization in the internal ventral structures. You got a liver, you've got a heart present here, which you cannot see in the Amphioxus. You've also got some differences in the brain region as well. You've got a little bit more enlargement and the difference in the extension of the notochord, right? The notochord is present in the ammocoetes, but it ends before the brain, right? That was a big distinguishing factor that we had talked about for the Amphioxus. So you've got, we're going to take it from the most rostral section of the lamprey, and we're going to move back. So you've got the oral hood, and that is surrounded by cirri, little tentacles, right? Similar to the Amphioxus. Behind that, you can see an eye, which you don't see in the Amphioxus. You can make out a little bit of an enlargement, which is their brain, and that extends down. Now we call it the spinal cord, okay? Just ventral to that, again, we've got the notochord running the length of the body. And then ventral to that, you can see the pharynx, followed by the esophagus, leading to the intestine. And then sort of just ventral to the esophagus, you can make out a little chamber where the heart is there, okay? So I'm gonna enlarge that so you can see that a little bit better, okay? Little heart there, and a liver present just caudal to that, okay? The intestine, of course, terminates at the anus, which is just before the postanal tail. Okay. All right. So now let's look at the ammocoetes in cross section. Okay. So we've got a couple of slides. You can see this is the most rostral section. It's cut through the eyes. You can see that there is the oral cavity here. This again is where it's going to have food and water entering the eye. On the dorsal side, now you've got this nervous system structure. This is their brain. You should note that you do not see a notochord, right? So you can see the brain is extending more rostral than the notochord. Okay, so all of this is a notochord. And then you can see some carotid arteries on either side, bringing blood to the head region, okay, from the heart. If we move a little bit more towards the tail, now we have the brain region, which had been nice and big here, is much smaller in diameter. So now we are going to refer to it as the spinal cord. It's no longer the massive structure, the glomerulus. So that mass of different structures. Now it's a spinal cord is much narrower cylinder. Okay, just ventral to that now is the notochord. And if you look at the picture, you can kind of see often in the notochord, you can see these little partitions inside there. That is a good distinguishing feature of the notochord. If you recall back from last semester, I do think it looks a little bit like adipose tissue that we looked at last semester. These little almost like bubbles in there, right? But that's the notochord. And then decrease that there. And then on the ventral side, we've got the pharynx now with the gill bars and slits in between. And along the outside of the organism, we've got musculature, again called myomeres. And that is pretty much all we can see now in this section. So we're going to move to the next one. You still have the pharynx with the gill bars on the ventral side. You can see the notochord again. It's got that sort of fragmented appearance to it on the inside. Just behind that, now it's kind of flattened out a little bit, but this is still the dorsal nerve cord or spinal cord. On both sides, you're still looking at a lot of myomeres, okay? And then in the position just ventral to the notochord, we can see the dorsal aorta, okay? So bringing blood from the heart along the length of the body. It's just going to be just ventral to the notochord, okay? And if you move then to the next section, most towards the tail, again, you've got the notochord, and in that same position, just ventral to that, you still have your dorsal aorta. Just dorsal to that, you still have your spinal cord, right? And around it, you've still got myomeres. If you look now, though, the abdomen region has changed, right? We no longer have the gills, so it's not the pharynx anymore. We now have an intestine, which is this section here, the intestines, and the kidneys, which are present, two of them on either side, okay? So in that region where the pharynx used to be, okay? And then we're also starting to see the dorsal fin appear here on the dorsal side. And if we continue more towards the tail, we have the fin extends into the caudal fin, which wraps around the tail on both the dorsal and around to the ventral side. Lots of musculature or myomeres. The notochord, the spinal cord is just dorsal to that. And then as you travel back towards the tail, that position where the dorsal aorta had been is now what we call the caudal artery, taking blood back to the tail, caudal artery. And then, of course, it will return through the caudal vein and return to the heart. Okay. There are some other blood vessels which the ammocoetes has that we can't always visualize in the slides, but you should be aware that the blood is in a closed circulatory system, which will return that blood to the heart. Okay. All right, so that is the last slide for the larval lamprey or ammocoetes. So now we're going to look at the perch. Okay, so the perch is a ray-finned fish. It's in the class Amphioxus. Okay, so it has a bony skeleton. And it is going to be another example of a vertebrate. Okay, so it has some external anatomy that you should be familiar with. It has several different fins we're going to look at. So first, it has an anterior and a posterior dorsal fin, number 1 and 2 here. Then it has a caudal fin, which is the fin on the tail there. Moving around to the ventral side, number 4 is the anal fin, which is just behind the anus. As you'll see, there's a pore here. That's the anus. And then number 5 is the pelvic fin. And number 6 is the pectoral fin. It's a little less clear, but they are present bilaterally. Okay. Other structures that you should look for in the perch before we actually cut it open are the operculum, which is this little flap here. It's almost triangular in shape. It's number 7. It sits right on top of the gills. It kind of covers and protects the opening to the gills. The lateral line, which is number 8, and I'm going to enlarge the picture to show you that a little bit better. The red line is just sort of indicating the rough area where that lateral line is. But if you can trace sort of my cursor here, if you can see that, you can see that there is this pale line that kind of runs down the length of the organism. Hopefully you can see that. These are a system of cells that detect vibrations in the water. Okay, so vibrations caused by movement of other organisms in the water. So obviously this helps in a sensory way with the perch's ability to perceive its environment, right? So if, for instance, a fish is traveling in a school, this lateral line system helps it to sense the movements of the others in its school. Additionally, obviously, it can sense the incoming predators if there's something approaching, movement of prey if it's feeding, right? So that lateral line senses detection or detects vibrations in the water environment. Okay, and then lastly, the maxilla and the mandible are the top and bottom jaws. Okay, so we've got, now we're in the jawed group of organisms. So we have a maxilla and a mandible that you can see here, top and bottom jaw bones. That is about it for the external anatomy for the perch, but we are going to do a dissection of this when you come back. So we're going to have you guys cut the abdomen wall away first. So you're going to start at the anus, which is about here, and you're going to cut along the midline up towards the head, and then cut dorsally to go around the operculum. You can remove the operculum, and then remove that section of the abdomen wall. Okay, so you're going to cut down here then, and you'll then be able to see a lot of the internal structures. We're gonna take a look at the more superficial ones, and then we'll have you guys remove those and see the more interior ones. So the first structure that you will probably notice in your perch will be the gonads. And you can tell the difference between a male and a female, again, based on the appearance of the gonad that you encounter here in this abdomen. It's going to be taking up a large amount of the space on the ventral side here. If it has a more off-white and sturdy structure to it, if it's more sturdy, it's likely a male. If you touch it, if you palpate the gonad and it comes apart readily, and you start to see little spheres, little balls, that is likely a female, because those are the eggs that then will be released for fertilization. So if you touch this area here and it crumbles very easily, it's likely a female. If it's a little bit more sturdy, it's likely a male. And sometimes for the females, you can actually see the eggs coming out through the urogenital pores and you'll be able to see the, in some cases, even before you cut the fish open. So you might be able to tell what you have even before you cut it. But if you do start the dissection, you should be able to tell based on the consistency of that gonad there, okay, whether you have a male or a female. All right, so that's taking up a large amount of the space in this region that you just exposed. You should also notice the gill with filaments, okay, which allow for oxygen to be extracted from the water for the perch. The liver should also be pretty apparent at this point. It has two flaps to it, okay? So there's a couple different flaps. It's this pale white structure, which are relatively thin flaps. You should be able to see that there. There's like a dark brown, kind of greenish spleen present here as well, which you should be able to see. And then if you continue and remove your gonads and remove your liver, you can uncover other parts of the digestive system. So you should be able to see the esophagus, which would be underneath your liver. You might have to remove the liver to see that. The esophagus, again, connects the mouth with the stomach, which is right here. So you should be able to see the stomach then as well, okay? And then there are also pyloric cecae, which are shown here. These help with digestion. You can also now see the swim bladder. It's going to be sort of up in this region here. It's going to be more dorsal than the gonads. So you can see this fine membrane here. You can actually cut it open as well. The swim bladder allows for the fish to position itself as far as changing depth by having different amounts of air or water in it. When you cut open the swim bladder, you should be aware that there may be a lot of fluid that comes out of it. So just be conscious of that when you're doing your dissection. Don't be surprised when you have a lot of leaking. All right, so we are going to then move to, oh, did I mention the intestines? Sorry, I should mention you can also see the intestines. So after the stomach, food will be processed through the intestines. There's a small and a large intestine. This one here is a large. You can see the small one also on the next picture here. So from the stomach, food travels to the small intestine, which is here, and then to the large intestine, which connects to the anus. Okay, so all the waste will then be removed through the anus. Okay, so additionally, if you continue to look, if you remove and look a little bit deeper, you can see the heart, which is present on the ventral side, little bilobed heart. It has one atrium and one ventricle in the perch, okay? And it is more rostral to most of the digestive structures, okay, that you'll see. And then lastly, we would like you to look for the brain. So obviously you're going to orient the fish such that you're looking at the dorsal side. Okay, so this is the dorsal side. This is the rostral end where you have the maxilla over here. And then of course, the fish is traveling lengthwise down this way. So you will remove some of the structures, the outer structures. You may have to work from the sides so that you don't damage the brain as you're cutting. If you want to try to remove the skull, that does take a little bit of work. It is a little bit tough, but if you are successful, you will see the brain, which has this enlargement, right, compared to the spinal cord. You can see their brain there, located between the eyes, okay? All right, so my last question for attendance, since I kind of forgot to ask additional attendance questions, is what structure is responsible for covering the gills, okay, in the perch? That will be my second attendance question. So there'll only be 2 this week. What structure covers the gills in the perch? Okay, so you should be able to answer that from our discussion. Okay, so additionally, for this week, there are 5 videos of microscope slides posted on Canvas for you to look at. Okay, so those are links to YouTube where you can see the cross sections and longitudinal sections of the Amphioxus and the ammocoetes. There are 3 of the Amphioxus. 1 is a longitudinal and 2 are cross-section slides because one's a male and one's a female. Okay. And then we have the longitudinal and the cross-section for the ammocoetes, which is the larval lamprey. Okay. And then, so view those, submit the 2 questions that I had given you as far as attendance this week. And then in person next week, be prepared for all the dissection craziness that we are going to do. So review all the material for the crayfish, the Nereus, the sea star, and the perch, which are going to dissect all in next week's lab. Okay. Any questions? You can always shoot me an email at cfry@lvc.edu. And I will see you soon. Have a great week.